There we go. All right. Good morning and welcome to Creating a Roadmap to Quantitative Systems Pharmacology. And we have a great program for you today in a very important and exciting area that will help us hopefully reduce some of the drug development time, and especially in this case, in the case of rare diseases. Next slide, please. So my name is Audra Stinchcomb. I am a professor here at the School of Pharmacy with Jim Polly, who is in charge of this MCERSI Center. We are an FDA-sponsored center at Baltimore and the College Park campus at the University of Maryland. And our goal here at the center is to develop, help foster the development of regulatory science in partnership with FDA. And it includes everything from research into developing new tools, standards, and approaches to assess safety, efficacy, quality, and performance of FDA-regulated products. Next slide. So I'm want to point you to the MCERSI workshop website for today. It has the full agenda and the full biographies of all speakers. We're not going to go into all the biographies because of our short schedule. And what's great about these meetings is within a few weeks, the presentations will be available along with the recordings for you to go back and reference at a later time. And there is the website address if you don't have that. Next. And for today, when you want to ask questions, make sure you use the chat box feature. We are not using the Q&A section of the Zoom meeting. We're using the chat box. So please make sure if you have a question, type it into the chat box and make sure it goes to everyone. If you want to ask an individual speaker, make sure you still send the question to everyone because someone else will be checking that chat box for questions. And then put the speaker's name in parentheses if you have a question for a certain individual. There's also closed captioning available today if you need it, and there's the directions there. Next. We have some other in-person workshops coming up, May 23rd and 24th. We have a drug dissolution and oral drug absorption workshop coming up. And then in August, we have physiologically based biopharmaceutics modeling, best practices. I would like to welcome our chairs of the meeting from FDA today. They've put a tremendous amount of work into this workshop and are very committed to this area of research and a path forward in drug development. And that is Dr. Bob Schuck and Dr. Jane Bai. They are members of the FDA, and you can see their bios here and more details on the website. But what I want to point out is we're a full-on Michigan team today. I received my PhD from Michigan. Dr. Schuck received a PharmD from Michigan and then a PhD from North Carolina. And Dr. Bai received her PhD from Michigan as well. So with that, I will hand this over to Dr. Schuck. Thank you.
Thank you very much, Audra, for the introduction, and hopefully we didn't lose any of our Ohio State friends on the line or anything during that, but we'll go ahead and get started. Let me share my screen. So welcome everybody to the FDA CERSI workshop on creating a roadmap to quantitative systems pharmacology informed rare disease drug development. As Audra mentioned, I'm Bob Schuck from the Division of Translational and Precision Medicine and FDA's Office of Clinical Pharmacology. And I'm very excited about today's workshop, and I'm highly optimistic that this will be the start of a conversation and a process to effectively use QSP to tackle some of the challenges that we encounter in rare disease drug development. I'd like to start out by thanking the organizing committee, including the co-chairs and all of the University of Maryland and FDA staff who have done an enormous amount of work to organize this workshop, as well as all of our esteemed speakers, panelists, and moderators. A special thank you to Jane Bai, who was really a driving force for this workshop from the inception of the idea to the submission of the CERSI proposal and identifying the speakers, and has really put a lot of enthusiasm and effort into making today happen. So to get into a little bit of the content that we plan to talk about today. So I think those of us that work in the rare disease drug development space are aware of the myriad challenges in developing drugs for small patient populations. So there's a ton of heterogeneity in the patient population, often spanning age ranges from infancy to adulthood, mild to severe phenotypes, a variable course of disease, whereas some patients might have a very progressive disease, others might have a less aggressive form of the disease, and many other sources of heterogeneity. We often have a lack of established biomarkers in these rare diseases that can be used for things like dose-finding studies and establishing safety and efficacy in clinical trials. And there's a simple small number of patients to gather all the data that we ideally want to see for these dose-finding studies, efficacy and safety studies, and other valuable clinical studies that are traditionally conducted during drug development for more common diseases where we have a larger patient population. Now, many of us that are in the clinical pharmacology field have also seen the utility of model-informed drug development approaches being used in recent years. So these modeling approaches have shown to be useful in optimizing clinical study designs, informing dose selection, informing disease progression, supporting the use of drugs in pediatric populations, and many other purposes. So hopefully you've noticed at this point that I've been repeating myself a little bit, and that's because there's a large amount of overlap between rare disease drug development challenges and the capabilities of modeling approaches. And therefore, today we're bringing together experts in rare disease drug development, as well as experts in quantitative systems pharmacology, to talk through some of these issues. And in session one, we'll start out at a high level, talking about rare disease drug development challenges, and hopefully identifying some of the challenges that are amenable to QSP approaches. Once we get into session two, we'll be talking about some successful uses and examples of QSP in drug development, and hopefully these can inform how QSP approaches can be used within rare diseases. And in session three, we'll be talking about how we can develop useful QSP models in rare diseases.
Hey, Bob, sorry
Yes.
to interrupt real quick. Your control bar is on the screen, and I don't know if you're changing slides, but you are currently on this first slide.
I'm not. I only have the introduction slide.
Perfect. Thank you.
Thanks for the warning. So without hesitation, let's go ahead and get into session one. That was actually a well-timed there. I'm the co-chair of, or I'm the chair of session one. I'll provide a brief introduction to the speakers as we get started, but as Audra said in the introduction, please see the list of biographies on the MCERSI website for additional information on each of our speakers. So our first speaker is Dr. Kerry Jo Lee, and she is the Associate Director of Rare Diseases at the FDA. And I'll hand it over to her for her presentation. Thank you.
Great, so hopefully you can hear me. Thanks so much, Dr. Schuck, for introducing me. I am Dr. Kerry Jo Lee. I'm the Associate Director for Rare Diseases and the lead for the Rare Diseases team. Our team is responsible for the PDUFA 7 rare disease commitments for the center, as well as serves as a program management office for CDER's new Accelerating Rare Disease Cures program. And I am really here to give you the 360-degree view of the progress that we've made, as well as the challenges that we still have to address in rare disease drug development. This is just my disclosure slide. And I'm going to get rid of that. So I'm going to talk a little bit about the overview of the progress that we've made in rare disease drug development, as well as the challenges and considerations in rare disease drug development. And then finally, really leave us with the message that collaborations are the key. Rare diseases are challenging, and it truly requires every stakeholder in drug development to come to the table and work together in order for us to overcome these challenges. So for an overview of the progress that we've made, we are seeing a lot of progress in rare disease drug development. Between 2015 and 2022, CDER approved 180 novel drugs, that's new molecular entities, for rare diseases. And of all the novel drugs, this is 50% of those that were approved during this time. In addition, since the passage of the Orphan Drug Act, the FDA has approved over 550 unique drugs and biologics for over 1,100 rare disease indications since the passage of the Orphan Drug Act. And however, despite the fact that we are making significant progress, there is still a lot of work to be done when you consider that approximately 30 million Americans are living with a rare disease, and the vast majority of these do not have an FDA-approved treatment. Another way to look at it, if you go back approximately 10, 12 years ago, starting in 2010, you can see the progress that rare disease drug development in CDER, the Center for Drug Evaluation and Research, has made over time. This shows the number of novel drugs or new molecular entities over this time period, and the columns are divided into the number of orphan drug approvals in green and the numbers of non-orphan drug approvals, non-rare diseases, in blue. And the purple line is the percentage of orphan drug approvals in each year. And since 2010, you'll see that the number of orphan drug approvals has risen dramatically in the United States. And in addition, the percentage of all approvals that are orphan drugs is consistently over 50% in 4 out of the 5 past years. So that's 54% in 2022. In addition to the novel drugs that you're seeing here, there's a number of drugs that the FDA approves, which are called supplemental approvals. So these are not novel drugs, not new molecular entities, but perhaps expansions to additional indications. And those are additionally continuing to grow. I want to highlight our use of expedited programs. These really help to facilitate rare disease drug development. They're intended to facilitate and expedite drug development to address unmet medical need in the treatment of serious or life-threatening conditions. Well, this is a vast majority of rare disease indications. The programs that we have are fast track designation, breakthrough therapy designation, accelerated approval, and priority review designation. And what you can see here is that predominantly rare diseases are the drug programs that utilize these expedited development programs. And what you get with them varies on the specific program, but it essentially means you'll get things like the ability to submit rolling review, or you can submit portions of the applications as they are complete in order to sort of facilitate review. You can have a faster timeline in something like priority review, in which the drug can be reviewed once submitted within a 6-month time frame instead of our standard 10-month time frame. And then also things like accelerated approval, which is a pathway by which you can approve a drug based on a surrogate endpoint that is reasonably likely to predict clinical benefit. And just in general, you see that the rare disease drugs truly do benefit from these expedited pathways, enabling us to marshal more resources and speed drug development in our ability to review these programs. Next, I want to just highlight some challenges and considerations in rare disease drug development. So although we have come far and there's a lot of progress being made, what we have seen is that in the landscape of rare disease drug development, there are common challenges. And the common challenges that we often face are that the natural history is poorly understood often. And this is hard because natural history is what we use to inform how we create a drug development program. We find that diseases are progressive, serious, life-limiting, and often lack adequately approved therapy. So this means urgent need. Many of these diseases have pediatric onset, approximately 50%. And there are additional considerations when you're working with a novel therapy in a pediatric population. These are very small populations. And what this does is often restrict our study design options. As well as study design options, it can make it very challenging to analyze the data that we do have, particularly when the therapeutic effect of a drug is modest. There are also phenotypic and genotypic diversity within a disorder that can make it very hard to sort of pick the correct patient population to enroll, particularly if the natural history of a disease is not well understood. Development programs often lack a solid translational science background, and this is critical in rare diseases. This the translational science aspect is so important, both to being able to choose the correct surrogate biomarker as a primary endpoint, if that is what you are doing, or to enable what most rare diseases do, which is follow the pathway to substantial evidence for effectiveness, where we utilize one adequate and well-controlled trial plus confirmatory evidence. And that confirmatory evidence is often strongly supported by the strength of your translational science. There are drug development tools that are often novel. So your outcome measure, whether it's a surrogate biomarker or a clinical outcome assessment, is often novel. These are first in indication programs the majority of the time. There's a lack of precedent therefore, which includes finding these clinically meaningful endpoints for drug development in many rare diseases. There's also common considerations in the environment for rare disease drug development. So many smaller companies with less regulatory experience work in this space. There are a lot of active patient stakeholder groups looking to navigate and participate in rare disease drug development. There's a dedicated academic community that may have limited knowledge of regulatory requirements or aspects of clinical trial development. And this is critical when you're submitting a marketing application for regulatory review. And so the conclusion is that we at the FDA really must engage our stakeholders in order to enhance their understanding of the regulatory process, as well as gain their alignment and support. We really can't do this in a vacuum. Collaborations, therefore, are really the key to rare disease drug development. And CDER this past year, almost exactly a year ago, launched the Accelerating Rare Disease Cures program. And I'm also going to talk about a number of other rare disease initiatives that we feel are really contributing to trying to advance rare disease drug development. The ARC program in and of itself was really started with the vision of speeding and increasing the development of effective and safe treatment options to address the unmet needs of patients with rare diseases. Our mission is to drive the scientific and regulatory innovation and engagement, because that conversation is key to accelerate the availability of treatment for patients with rare diseases. How the ARC program is organized is that it is governed by senior leadership from those super offices that are closest to regulatory review. So that includes the Office of New Drugs, which is where the clinical offices, as well as the pharmacological toxicological offices exist, as well as the Office of Translational Science, within which the Office of Biostatistics and the Office of Clinical Pharmacology are located. And then the Office of the Center Director, because of the importance of this initiative, is also very, very involved. The Program Management Office for the CDER's ARC program is the Rare Diseases team. Now, just because these are the offices that govern the program does not mean that we do not work across the center. CDER is vast, and we have to work with our partners in the Office of Medical Policy, which is where real-world evidence, real-world data is located, and the Office of Surveillance and Epidemiology, who also plays a role in the evaluation of that evidence, as well as post-marketing safety. The Office of Strategic Programs, where our benefit risk considerations and decision science rest, and other CDER offices and divisions as we need them. We additionally continue to partner with the other FDA offices and programs, particularly those that work on similar indications, such as the Center for Biologics Evaluation and Research and the Center for Devices and Radiological Health, as well as the Office of the Commissioner, the Office of Orphan Products, the Office of Pediatric Therapeutics, and the Patient Affairs staff. And we also do work with the Oncological Center of Excellence very, very closely, given our overlap in rare disease indications. Just a few words about the commitment from the Office of Translational Science, under which this endeavor and workshop comes. You know, as I spoke before, translational science is incredibly important to rare disease drug development. It's the field of investigation which seeks to understand the scientific and operational principles underlying each step of the translation process. And just here you can see a number of programs under the Office of Translational Science, with which they work with the other super offices and collaborate, such as precision medicine, rare diseases, drug development tool qualification, the MIDD and CID programs, real world evidence, and the patient voice. Each of these contribute to our ability, our regulatory science capacity, our regulatory review capacity and expertise, the guidances that we put out, as well as our own policies and procedures, standard operating procedures and review tools, stakeholder engagement, pilot programs such as the CPIM, and knowledge management and communication. Specifically, the Office of Clinical Pharmacology, and you'll be hearing from our leadership here during this conference, works to drive the development and use of the regulatory and scientific tools. This is really what they bring to the ARC program. They work on platforms to facilitate natural histories, methodologies to construct novel endpoints, drug disease modeling, dose selection, and that really critical component of translational medicine. And then stakeholder engagement, again, I really want to underscore We have to have bidirectional communication with our stakeholders so that everyone understands the regulatory framework within which we work to facilitate the best packages that we can in order for us to make determinations on whether or not a drug is effective. I'm going to start skipping because I don't want to go super long. But our first year, we focused on engagement in the ARC program, and we did this for two reasons. First of all, obviously, to share what we could about the regulatory framework to help stakeholders, but secondly, so that we could hear from stakeholders about what the challenges were that they had in constructing regulatory programs in order to submit for evaluation in rare disease drug development. And we've held a number of conferences, which I have listed here on a variety of topics. And the idea is that these live on in perpetuity and serve as informational sessions for people looking to learn more about these aspects of drug development. So if you were to go to the CDER ARC webpage, you could find links to each of these conferences. And they were very, very valuable resources. I also want to highlight, as part of what we're doing in CDER ARC, we're really trying to work to promote knowledge sharing about rare disease drug development. And therefore, we have FDA Track, which is an agency-wide performance management system that monitors key performances. And one of those is how we are doing in terms of marketing applications submitted and approved. And what we have done is add a rare disease filter to this tracker starting in 2023 so that people can track CDER's progress in rare disease drug approvals. I want to speak a little bit about the learning and education to advance and empower rare disease drug developers, which is another initiative launched under ARC this past year. This was really an initiative where we did a lot of outreach to seek input from stakeholders who design or conduct rare disease drug development programs. We opened a docket, which recently closed last month, and heard from people about what they identified as their knowledge gaps for stakeholders about the regulatory process of rare disease drug development. We will take this information to create or expand educational resources for stakeholders. So from our standpoint, we wanted to better understand the challenges that stakeholders have in bringing together a regulatory drug development program. Given that we hear from people on what they believe their gaps are in the areas of non-clinical and clinical pharmacological considerations, clinical trial design and interpretation, and regulatory considerations, we really want to develop further educational materials on these fundamental topics that will be available to external rare disease drug development stakeholders. In parallel with this, our patient-focused drug development group is working with the National Organization for Rare Disorders to work on a patient toolkit educational series. And we're hoping that this is really valuable to stakeholders to help them, given how many new companies we have in this area, as well as academics. If you want to hear more about these, our workshops and other initiatives that we launch under the ARC umbrella, please subscribe to it's just a plug to subscribe to our listserv. I also wanted to talk about an initiative that's been going on for several years, but may not be known well. It's the Rare Disease Cures Accelerator Data and Analytics Platform. This is important because rare disease populations are rare. Splitting the information that we have on populations really slows down rare disease drug development. So this is an initiative between the FDA CDER, Center for Drug Evaluation and Research, the Critical Path Institute, and NORD, where you are able to submit clinical trial data, registries, natural history data, genomic data, imaging data, surveillance data, EHR data, and other data into a data vault that will curate this data and make it available for analysis across indications. This is open to all, it is secure, and we really hope that by people being able to utilize this, they can learn more about the natural history of conditions in order to inform very thoughtful drug development programs. We want to share this data because, as I've said, rare disease data is rare. Every piece of it is incredibly valuable. People who take part in these studies want their data used. They don't want it to just go away. They want to help develop drugs that can help their conditions. Everyone is able to use this. Combined data is incredibly informative, and data really needs to be shared in a responsible way that enables it to be both interpreted and utilized well. I also wanted to take a brief moment to highlight the ARDEA program under PDUFA 7, the Rare Disease Endpoint Advancement Pilot Program. This is a program that is a joint Center for Drug Evaluation and Center for Biologics Evaluation Research program. And it's going to help to advance rare disease drug development by allowing additional collaboration with the FDA for the efficacy endpoint development progress. As I said, these are often novel drugs in new indications that don't have therapies, so an endpoint is novel. There's a lot of challenges in utilizing an endpoint for the first time, particularly in rare disease trials where we don't often have phase 1, 2, and 3, but in fact, we have sort of a seamless design or one bite of the apple in order to develop a successful drug development program. So this is open for business next month, or actually no, in 2 months, in July 1st. And the associated drug development programs that can be submitted to this should be active and address a rare disease with an active IND or pre-IND, with the exception of potentially natural history studies that are intended to develop a novel efficacy endpoint. And the endpoint should be novel, so it has not been used before to support a marketing application approval in a particular indication to establish substantial evidence of effectiveness for a rare disease treatment. This is a pilot program, so it's quite small. There will be 1 proposal accepted this year and up to 3 in subsequent years of PDUFA. But what we will be doing is sharing the learnings through this program in up to 3 public workshops. Again, we really believe that informing stakeholders of learnings and how to best construct, endpoints or design trials in rare diseases is one of the most important things that we can do. The goal is to promote innovation and evolving science. So there is now a guidance that will be coming out at the end of this program on developing novel endpoints, as well as I said, the 3 workshops. That being said, there's one coming up in a month, which you are welcome to register for if you'd like to learn more about this program. And finally, one of the things we learned through Leader 3D already, we have a lot of guidances out there, and a lot of them people don't know about. So I'm just highlighting a few here that are critically important for enhancing diversity, as well as utilizing natural history studies for rare diseases, common issues in rare disease drug development to look out for, and a recent one of the Real World Data, Real World Evidence program about the design and conduct of externally controlled trials for drug and biological products. These are all very important guidances, and I just wanted to highlight them here. I have an additional page of just some other fun ones. Registries is also critical. We get a lot of good data to inform trials from registries. And as well as N of 1 therapies. So if you don't have a drug development program for a product, but you are treating 1 patient or a handful of patients, particularly with the antisense oligonucleotide drug products, we have a number of guidances out on how to best do that I thought would be helpful to share with the audience. So in conclusion, rare diseases are challenging. There's a lot to be done, and engaging with us early and often and at as many of these conferences that we are trying to go to as possible can be really, really helpful. And CEDAR has a number of programs by which we are trying to facilitate and advance rare disease drug development. Thank you.
Thank you very much, Dr. Lee, for the excellent overview of some of FDA and CEDAR's efforts in the rare disease drug development space. I do believe we have time for a couple of questions if anybody has them to post in the chat, and if not, then we can move on to the second speaker.
I think I saw some, and I can work on addressing those. People wanted a few things posted in the chat.
Any questions for Dr. Lee? I see one coming through, a question on how do you engage ARC as a sponsor? You're on mute, Dr. Lee.
Thanks so much. That's a great question. ARC in and of itself does not perform regulatory review. Regulatory review goes through the divisions in the way that it always has. In terms of engaging us through as a sponsor, we, all the workshops that we do, the Leader 3D programs, the engagement that we're working on, that is sort of how we work. We also speak at a number of events. I've been contacted by a number of groups, as has my team, just to promote our learnings. What we really are is an umbrella for CEDAR's rare disease programs, and our focuses are really engaging with stakeholders internally and externally in order to drive the advancement of key priorities.
Thank you, and I see a few more coming through now. We have maybe 1 or 2 more minutes. One says the breadth and depth of programs related to rare disease drug development is impressive but overwhelming. Do you have the equivalent of an ombudsman to help one navigate the systems?
Yeah, so this is also a great question. This is one of the things that we've been learning through Leader 3D, just through the feedback that we are getting. And really, because we're seeing ARC as the umbrella, we're really going to be working hard on our webpage for that to become a hub for people to go to, by which they can launch to, critical guidances, to critical other rare disease endeavors going on across the center. So this is something that we're going to be working on in year 2, given feedback such as we've heard from you, as well as through the other engagement that we've done. Because it does no good if we're sort of throwing information out into the void and no one is able to find it. So that is definitely a focus for us.
All right, thank you very much. I see a couple more questions, but I think we're running a little bit short on time. And unfortunately, won't be able to get them all, but maybe you can revisit some of those during the panel discussion. At this point, I'd like to thank Dr. Lee and introduce our second speaker, who is Dr. Issam Zineh. Dr. Zineh is the Director of the Office of Clinical Pharmacology at FDA, and he'll give a presentation titled The Quantitative and the Mechanistic Strategies to Advance Disease Drug Development Through Regulatory Science. So thank you very much, and I'll hand it over to Dr. Zineh.
Thanks, Dr. Schuck. Can you, just a tech check, can you see everything clear with no obstructions, and can you hear me?
Yes, we can see and hear clearly.
Okay, well, thanks very much to Dr. Schuck and Dr. Bai and all the organizers for the invitation and for bringing us together on this important topic. What I was hoping to do in the next few minutes is to put QSP into this broader context of model-informed drug development and highlight some of the considerations to date in the application of QSP from a regulatory perspective. And what you can see here is that we have a long history in this space of MIDD. Model-based strategies have been long envisioned by FDA as well as other regulatory bodies as an approach that could reduce attrition, also streamline development through obviating the need for some non-clinical and clinical studies, and to serve as a bridge in cases of regulatory uncertainty. And we've endorsed MIDD strategies in numerous ways to advance safety, efficacy, and quality of therapeutic products. The problem historically has been that these approaches have been opportunistically applied. They've been heterogeneously accepted across a product life cycle or across different therapeutic areas. And there had been no dedicated pathways for engagement, direct sort of engagement with subject matter experts within the context of a given development program. So we've made some specific investments to bolster MIDD. Dr. Lee mentioned a few of them, and these have really been intended to circumvent or overcome some of these historical challenges, and we'll touch on them a little bit. So where are we in terms of the promise and application of QSP? I think that the audience appreciates that many scientific innovations, including regulatory science innovations, have kind of followed this classic hope-hype cycle in which early on there's this combination of some scientific advancement and community interest that leads to really high expectations for the science to address some unmet medical need. These expectations can become outsized relative to what the science can deliver. And so you get this peak of inflated expectations. And then that's followed by this trough, which is really driven by either the innovation failing to deliver the magnitude of the promise that was originally envisioned, or I should say, or failing to be implemented as expeditiously or as smoothly as envisioned. But then through continued investment and through champions and accrual of success cases, which is really critical, we get to this plateau of productivity or this mainstreaming of the science into, for example, decision-making, which is an aspirational place to be and a very good place to be. Now, on the right, you see this exponential growth of activity and interest in MIDD generally and QSP specifically, if we're using publications as sort of a crude proxy. So this begs the question of where we are on this hope-hype cycle. Are we still on this upswing of enthusiasm that will eventually be recalibrated, or are we on this slope of enlightenment in which decision makers, scientists begin to understand the space in which QSP can have utility, including the boundaries within which we can comfortably make decisions? You can see on the left-hand side of this slide where we're seeing growth in the estimated number of drug applications submitted to the FDA that include a QSP component. And we'll need a few more years of data, but it looks like we're reaching a steady state. On the right, you can see that QSP has been applied across many therapeutic areas, the most common and descending order being oncology, representing nearly half of the applications we see, followed by infectious diseases, endocrinology, neurology, and this category called rare and genetic disease, and then others. And I want to note importantly that this category of rare genetic disease is mostly comprised of inborn errors of metabolism. So it doesn't fully represent all the rare diseases we see. These other areas like oncology and neurology also include rare disease applications. So this 3% here, I'd venture to say, is a number that doesn't fully account for all of the rare disease applications that we're seeing that have employed QSP. So we have real signs of progress over the years. How does QSP fare relative to other, what were once considered emerging regulatory sciences, including exposure response modeling, PBPK modeling, precision medicine? I've plotted these all along this hope hype cycle here on the right. And a few years ago made this prediction that QSP would, one, cycle through this hope hype cycle a lot faster than other regulatory sciences, and two, that this peak of inflated expectations wouldn't be as high. And I think that's borne out to a large extent. When we think of the historical barriers to translation that could affect this trajectory, they include things like constraints of the science itself, a steep learning curve among non-technical experts, coupled with very few instructive cases, and this high organizational activation energy required to integrate new approaches into very well-established workflows. In my view, the QSP community has really been generally proactive and thoughtful about these challenges. When you look at the literature, it's replete with examples of survey results, editorials, use cases that engage on questions of where the biggest impact is across the discovery and development continuum, what technology needs are required to advance application, what are the best practices in communicating model context of use, model development, verification and validation activities. So all in all, I think that this sort of prediction of this trajectory is on point. As Dr. Lee previously showed, there are several regulatory decision tool programs we've instantiated under previous versions of the Prescription Drug User Fee Act that we've carried forward and matured to present day. I won't cover all of these, but I just want to say that these were developed not necessarily focused on the rare disease space, but they're clearly of relevance to rare disease drug development and articulate pathways that include how to incorporate patient voice and caregiver voice into drug development, how to qualify biomarkers, have more structured approaches to risk-benefit assessment, and of course, what we're interested in today, employing model-informed drug development to enable more efficient and more informative clinical trials. And in the next few slides, I want to focus a little bit about our coordinated MIDD activities, because I think we've learned a lot from those programs, and the lessons learned are germane to QSP. Over decades of trying to integrate quantitative and mechanistic approaches into drug development and regulatory evaluation of therapeutics, we've concluded that to be successful in doing so basically requires three things. One is creating an environment that increases stakeholder acceptance of MIDD approaches, including among non-technical experts who are involved in the decision-making processes. Second is developing standards and best practices that lead to consistent application and evaluation. Third is increasing capacity and expertise to address growing demands in innovation. And again, Dr. Lee touched on some of these. I want to mention the importance of this stakeholder engagement component, we've done this through a series of workshops to identify best practices in a variety of MIDD areas, including in physiologically based pharmacokinetic modeling, design and analysis, and inference from dose exposure response approaches, disease progression modeling, so all sort of clearly relevant to what we're talking about today. We also stood up a pilot program on MIDD, which was a forum to engage more directly on these issues in drug development. And I want to talk a little bit about that program next, because I think it's been a huge success. And again, the lessons learned have been invaluable for QSP and other mechanistic and empirical approaches. So this program is essentially, at least the pilot was administered jointly by our office, the Office of Clinical Pharmacology and the Center for Drugs, and our colleagues in the Center for Biologics. It's a dedicated forum for regulatory interaction on MIDD application in a specific drug development program. Our three main priority areas or domain areas based on past experience, expertise, precedents were in dose selection and optimization, clinical trial simulation, and mechanistic safety evaluation. Although I'll say that these are not rigid. This program allows for conversations beyond these three application domains. And also it's important to say that this engagement can occur anywhere along the drug development continuum, from the early translational space into the first in human and early clinical development through late stages of clinical development. And we've had examples of that in the program. This slide is meant to illustrate the high demand for such a direct engagement. We were expecting about granting about 8 requests a year, at least in CEDAR. And you can see from this slide over the 5-year period, we've greatly exceeded that number. And this really speaks to the need and benefit of direct engagement on MIDD issues, including on QSP-related questions. So tremendous effort, but one that has been hugely successful. We saw this as a pathway used for gaining alignment on many of those specific topic areas in those three domains that I mentioned. And when we look at impacts, this was a paper published by some companies in the IQ consortium who participated in the program, and they report significant reduction in clinical development time on the order of months to years, fewer patients needed in clinical trials, fewer clinical trials needed, more expeditious drug development by informing go, no-go decisions. In terms of cost, some companies estimated that they saved up to $70 million per development program that went through this MIDD pilot program. And the companies also report alignment and clarity on a variety of science policy, technical and regulatory issues. And this is really driven by our core principle of providing very clear, very transparent regulatory advice on these issues. Now, one of the advantages of having such a coordinated effort and a dedicated forum is that through a knowledge management component, we're able to accrue understanding in what the key questions are, what the key issues are, what the recurring themes are in the evaluation of specific quantitative or mechanistic strategies. For example, we've had several interactions related to QSP approaches. They've mostly been used to inform a variety of drug development decisions in the clinical trial planning space. And what you might imagine, the conversations in these development programs between FDA and companies really focus on exactly the same questions that the broader QSP community engages on, namely around the knowledge model. Does the sort of conceptual framing of the biology kind of make sense? And around the computational model. Now, in terms of sort of the key questions in these around the knowledge model and the computational model, with respect to the knowledge model, the overarching question from the regulatory perspective, again, is not that different from the scientific perspective. Does the model, as proposed, capture the relevant aspects of the relevant biological processes? So this then becomes a question of model breadth. Are the relevant pathophysiological processes, drug disposition pathways, target engagement and downstream pharmacology, and if relevant, relationship between these pharmacological effects and clinical outcomes adequately represented? Is there a clear mapping between the literature or experimental evidence being cited and the assertions being made by the model? Is there explicit, transparent, objective rubric for weighing the strength of evidence that supports these assertions? Are the uncertainties and the variability and the plan for addressing the impact of these uncertainties and variability on interpretation clearly articulated? With respect to the computational model, the questions include whether the sources of the model parameters and assumptions are clear and credible. Is there a clear approach to model calibration, verification, and validation? I'll use these terms interchangeably. Whichever term of art we'd like to use here doesn't really matter much to me, but essentially the question is, how can we be confident that the computational outputs can be relied upon to inform the decision at hand? So to that end, these questions around the adequacy of the knowledge model and the computational model really should be framed in terms of a key question or set of questions that are being addressed, a key decision that can be articulated. We found that any discussion of the actual QSP sort of technical model specifics, or really the specifics around any modeling and simulation strategy, must be preceded by a discussion around this high-level question of interest, as well as the context of use, meaning how the model, along with other data and information, if relevant, will be specifically used to address that question of interest. We've also found that it's helpful to have discussions around model influence. And by that, we mean, how is the output of the model fitting into the totality of evidence? Is it a nice to have? Is it supportive of other data sources and information? Or will it be pivotal? Is it the key piece of evidence that will drive the decision? Decision consequence is another important dimension of these questions. And that is, what is the impact of making the wrong decision based on the available evidence? The benefits of having these questions and answers laid out is that it aids in efficiency and acceptance. If there's not clarity on these issues, we may default to the most conservative stance and be really reluctant to accept modeling and simulation, or this could lead to this kind of undesirable back and forth protracted dialogue between regulators and drug developers on what is the question, what are the data requirements needed to establish model credibility. All of that becomes very murky if we don't have a clear picture on the question that we're trying to answer. Regarding model credibility, articulating the model influence and decision consequence is very helpful. I would also add that understanding if there are any future uses of the model is an important consideration. One of the big challenges for us in the evaluation of QSP and other mechanistic models is that there could be an evolving context of use throughout development. For example, a model may be used initially as a clinical trial planning tool early in development. It may then be iterated for later use in development with the objective of replacing a clinical trial altogether. These are obviously very different intended uses. So if there's some line of sight on what future uses of the model might be, this could be very helpful in guiding early conversations on what set of activities and by what criteria the model would be deemed fit for its intended purpose. For example, questions around what the model needs to reproduce and in what population of interest. So what's next for QSP? This is just some text from this 2019 paper that I put out there. And at the time, my view was that the biggest return on investment of QSP was really in the drug discovery and the development space, not so much in the regulatory decision making. And so the growth and success of QSP would really depend on companies and their capabilities and their sort of culture of acceptance around these approaches, and that it really would not be driven by kind of the regulatory side of the house. And I think I definitely have changed my view on that as we see more QSP being applied for regulatory purposes. There's still an open question about how regulators should evaluate these models. This is these are complex and very elaborate and very time consuming. And so there is that open question of how we evaluate these models when they're being proposed for a variety of regulatory purposes. Also, I asserted that academic researchers, their role in this space should not be underestimated, given that the fundamentals, the mechanistic knowledge that drives model building really derives from basic translational and clinical research. And that's as true now as it was when I said this. So in terms of the specific opportunity space for rare disease, on the left here, I've listed some opportunity areas for QSP. The fundamental issue I think you heard Bob and Kerry Jo mention, and I think we all appreciate, is that in rare disease development, we by definition have exceedingly small numbers of patients. And so we often essentially have one chance to demonstrate clinical benefit. This is made more challenging by heterogeneous disease course, multi-system involvement, animal models that may not necessarily fully recapitulate the human disease, an absence of a single biomarker that's easily measurable and robustly associated with clinical outcomes. And so to that end, potential applications of QSP include in the clinical trial design space, around patient selection and enrichment, and around trial duration, dose selection going into clinical studies, as well as dose optimization in a sort of post hoc data-driven approach, biomarker selection, understanding biomarker dynamics and relevance to clinical outcomes, what I call strength of hypothesis. Oftentimes, again, we are limited in the clinical data sets that we have. And so we have to have confidence that any movement on a pharmacologically relevant biomarker or on an intermediate clinical endpoint is likely to translate ultimately to clinical benefit. And we don't often have that link. And so having a strong sense that the hypothesis is sound gives us more confidence when we have data that may be equivocal or of small effect. Clearly, QSP can be important in kind of being informed in how we look at safety. There may be a role for QSP in extrapolation, although I don't think the big challenge in QSP is extrapolating efficacy to unstudied populations. It's demonstrating efficacy in the first place. And then also this idea of reverse translation. Can we go from the clinic back to the bench? Can we understand the determinants of response variability, either things like variable dose response or non-responders, responder differential outcomes, and can that inform drug development in a kind of a reverse translational way? So I was very interested to see this paper recently published from the ISOP QSP scientific interest group. Some of the results are on the right here. They surveyed the attitudes among pharma, biotech, consulting, and academic scientists on QSP's impact, challenges, and future directions in the immuno-oncology space. And I really like this paper because it illustrates nicely the kind of work that I mentioned previously that the QSP community is doing to progress through the hope hype cycle toward integration. And also there's some parallels between immuno-oncology and rare disease. QSP is fairly nascent in those domains. Those therapeutic areas have some related challenges, for example, around preclinical animal models and around heterogeneity in patient presentation. So in the immuno-oncology context, you can see that the areas in which QSP has been applied include most commonly dosing, evaluation of combination therapy, clinical trial design, and biomarker identification. So not terribly different than what I've listed for rare diseases on the left. I also want to point out that the survey looked at response rates stratified by QSP modelers, non-QSP modelers, and non-modelers. And it's really interesting to note here that the authors note this as well, that there's discrepancy between QSP modelers and others on where QSP has been utilized. You can see that QSP modelers tend to be more positive about QSP's contribution almost invariably across these application areas. And in some cases, for example, biomarker identification, which is really critical in the rare disease space, it was a huge discrepancy compared to non-QSP modelers and non-modelers. So it may be worth thinking about this disconnect as we move forward in the QSP community in general and in the rare disease space in particular. Now, talking a little bit about challenges, this same survey reported that the #1 challenge for QSP, again, at least in immuno-oncology, but I think this is highly portable, is insufficient data to develop a QSP model. This could mean the data don't exist. It could mean that they're not easily accessible, either organizationally or even when they're presented in the literature. It's very hard to abstract out the needed information. The problem is compounded when we're talking about unprecedented targets, which is often the case in rare diseases. And this could represent a real opportunity for collaboration among the rare disease community. The second greatest challenge it cited is the relatively long timeline associated with QSP model development. This is driven by the fact that we're trying to represent complex biology, which we often don't fully understand. So to that end, there's often this very extensive, time-consuming literature review and iterative discussions with domain experts to inform the model structure, for example, the knowledge model. And I think it's very important to appreciate that when the use of QSP is proposed to the FDA, we often have to do the same arduous, time-consuming task of going back to the literature and evaluating the applicant's proposed model for face validity, the specifics of the model's assumptions, the robustness of those assumptions to variability, and so on. And so, and we have a much shorter time in which to do that exercise. We also don't have unlimited easy access to domain experts when you consider how many rare diseases we actually see. So these challenges that the community is seeing in terms of developing these models for drug development purposes, we also see on the regulatory side, and with even more compressed timelines. The other challenge identified in the top 3 by the survey is this challenge of model validation, which is often of principal concern when we're talking about using simulated results for clinical decision-making. These are usually data considerations, but the lack of specific regulatory criteria for model evaluation is sometimes cited as a challenge. From my perspective, it's going to be very hard to define a standard set of model evaluation criteria in terms of performance metrics. But I think that framework that I provided earlier around the question of interest, the context of use, the model influence, and so forth, gives us the necessary transparency to facilitate the conversation and is also adaptable to the wide range of potential QSP applications. So I just want to end by emphasizing that MIDD has nicely matured and is enjoying routine application. There's greater openness to a variety of modeling and simulation strategies, including mechanistic ones like QSP. QSP, in our experience from what we're seeing, has been mostly routinely applied in clinical trial planning in the rare disease and also in the common chronic disease area. And clarity on questions of interest and context of use is critical to discussions around whether the model is fit for its intended use. There are challenges inherent in rare disease drug development that make these kinds of mechanistic approaches very desirable, and the broad opportunity space exists, but there's a variety of challenges that are not specific to rare diseases, but that are broadly applicable and relevant to QSP that must continue to be addressed. And the only way to really do that efficiently is to do that through community and through collaboration. So I will end there. Thanks for the attention, and I'll turn it back over.
Thank you very much for the excellent presentation, Dr. Zineh. I think I did see a few questions in the chat, but I think we're at time, so perhaps we'll move on to the next presentation and maybe move some of those into the panel discussion. For our third talk in this section, I'd like to introduce Dr. Markus Ries from Heidelberg University Hospital, and he'll be talking about supporting orphan drug development with retrospective quantitative natural history modeling. I'll turn it over to Dr. Ries. So maybe while we wait for Dr. Ries to get back connected, we can take some of the questions that had come in during the last session. So I've got a list here, and I don't think we'll have time for all of them, but Dr. Zineh, one of them that came through was a comment that QSP modeling is time consuming and costs a lot. Getting the pharma company to invest is an uphill climb. What are your thoughts on how we can support the return on investment? And it goes on to say that there's several small companies in the rare disease space who have limited understanding of MIDD. Any thoughts on this?
Great questions. I mean, I think part of the answer to the first part of the question will hopefully arise out of today's conversation. But I can tell you that in terms of the return on investment in MIDD more broadly, and I think this is applicable to QSP as well, it wasn't, we as sort of advocates and evangelizers of the science have been advocating and sort of anecdotally talking about the return on investment for years. I would say that the work that was done by the IQ consortium that actually put metrics on what the return on investment is, whether we're talking about, clinical development time, cost savings, expediting go, no-go decisions, these kinds of things that really matter from the standpoint of either the economics of drug development or efficiency in drug development, bringing drugs to market quicker, or as importantly, killing compounds, earlier. Those really have been very important, even within FDA, to sort of raise the visibility of the impacts of MIDD on the kind of regulatory evaluation side. So I would encourage, if there are consortia out there or individual companies out there that aren't doing this already, to consider trying to derive impact metrics, even if it's anecdotal and kind of narrative summaries of the experience that go a very long way to making the business case, if you will. The second question was around, smaller companies around MIDD. Yes, also another great question. We have some resources. We have kind of an MIDD central database that at least we can share, website that we could share, a hub for people. We could do that as an after meeting action item or drop it in the chat. But there are also many academic institutions that I think you'll hear from a little bit later today that also have, educational initiatives or consultative capabilities that can help smaller companies along. I would say we're also very open. We tend to be very proactive at FDA about identifying pathways in which MIDD can help in a drug development context. And so if smaller companies want to come in, they're more than welcome to either use the MIDD pathway, a program pathway, or even the sort of typical IND pathways that exist, and request meetings with the interdisciplinary team, and we can work through that. We can work that way as well.
Thanks very much for the answer. And I see Dr. Lee is also on the screen. I have a question queued up from her presentation that actually, or that came in during her presentation that might be appropriate for either or both of you. So it asks, which guidances are most relevant to optimizing dose, given the past work practices that often require larger population for randomized phase 2 studies, et cetera? So any thoughts on whether there's FDA guidance out there or other documents that might inform optimizing dose?
I guess that's a tough question to answer, because when we say optimize, well, yes, there are. There's the exposure response guidance, which is being revised and probably soon to hit the streets, I would imagine. There's the POP PK. These are, of course, PK related guidances. So there are guidances that already exist around dose selection going into studies and dose optimizations. I'm trying to think of, within the context of QSP, there's not guidance around this per se, but I will say the majority of the QSP applications that we've seen have been, the clear majority have been related to dose, mostly around how do we mitigate risk. So I think it would be hard to say, you know, there's a one size fits all guidance. It's a constellation of guidances to look at. And then again, some of these are should be contextualized to the individual development program. If there is not a huge patient pool from which we can draw, you know, patients to empirically test many doses, we may need to use alternative strategies. We've approved, for example, or endorsed MCP mod as a statistical strategy that helps with not only the question of dose optimization, but multiplicity testing, which is very important when you're having smaller trials that will be, you know, serve two purposes around dose optimization and providing evidence of effectiveness. So I know I'm answering it broadly because it's a broad question. So hopefully at least this provides a starting point of what to check out.
Thank you. And Dr. Lee, did you have anything to add, or?
Sorry, I don't have a lot to add. I mean, Issam's really covered, I think, what's particular from a clinical pharmacology standpoint. I would also just maybe point out that when you talk about the broader context of the trial, you know, the adaptive designs for clinical trials guidance does talk about dose and incorporating that into adaptive design. And sort of when I think when you're stepping back and looking at it at the broader lens, that can also be one that might be helpful.
I said thank you both for the insight and directing us towards these helpful resources. I just received a message that I think Dr. Ries is back on, so we'll maybe try to turn over to him quickly for his presentation.
Excellent. Thank you very much for the kind invitation to this very exciting seminar. And I've already learned a lot by just listening in to your excellent presentations. I want to present a method which is called retrospective quantitative natural history modeling. I hope I'm not going to inflate your expectations. I think this method is a piece of the puzzle in the overall picture of understanding the natural history, and it potentially could support orphan drug development. So this is an overview of my conflict of interest. As I said, this is an overview of my conflict of interest from the past. And here is an overview of the talk. I'm going to speak about 4 seminal research questions that you can address with this particular method of quantitative retrospective natural history modeling. I'm going to speak about the limitations and the opportunities of this method in the overall picture. I'm going to present you with one practical example, how this model has been applied, and then I will conclude and follow with some perspectives. So if you go into natural history studies, you want to learn about the disease, and there are lots of questions that you potentially have, but with this particular method, I think you can nicely address these 4 ones here. So first of all, and this is probably more relevant to clinicians in the field or to disease awareness, how long does it take to make the diagnosis? Because you know, sometimes you need some sophisticated technology, some sophisticated tests that may not be available. There may be some barriers, or just people don't know the disease because it's so rare. Second, and then I think this is a particular strength of the method, is to give you some answers about survival. So how long do patients live, which is important for drug development, but also important for patient counseling. It's very important, you know, for parents to have this information, but there's always some sort of variability here in these numbers, so you need to be careful. Then it can tell you something about the disease outcome. So for example, the question is, can we identify any factors that are associated with a mild or severe disease in a particular condition, depending, for example, you know, from the genotype or particular signs or symptoms, or depending from biomarkers, are they associated with longer survival, for example? Then a fourth point, which is important and can be nicely covered with this method, it says you something about the epidemiology. If you want to recruit patients into clinical studies, you want to know where do they live, or if you're dealing with particular populations that are coming from one or the other part of the world, you know, it may raise your diagnostic suspicion if you see a certain combination of signs or symptoms and could point you to the diagnosis. So this is an example about this natural history project that we've conducted here at the University of Heidelberg, where I'm a pediatrician and pediatric neurologist. I've worked on lysosomal storage disorders, which are listed here, and then also in a epileptic epileptic metabolic epilepsy called molybdenum cofactor deficiency. As you can see that these diseases are rare or ultra rare conditions, you can see the numbers here with estimated prevalences or incidences in the literature taken from the Orphanet report series that publishes epidemiological data on a regular basis. And these are the numbers of patients that we could identify from the literature. How that works, I'm going to tell you that later, and which was very interesting. I thought that, for example, on galactosialidosis, 100 cases were the estimated prevalence or incidence, and we could identify 142 from the literature. So you're dealing with double or triple digit numbers here, but overall small populations compared to big drug development programs in more frequent disorders, but still a substantial numbers. I don't know if there are any, you know, clinicians, that there are a few out there, but who see like 248 patients with Krabbe disease. These are interesting numbers, I think. And the method is described in this paper that we've published here in the Journal of Inherited Metabolic Disease, which is called Quantitative Retrospective Natural History Modeling for Orphan Drug Development. If you want some background information, you're invited to have a look at this paper. So the method, how do I explain the method to students? Well, I ask them, you know, show them the picture and say, what do you see? And usually people say, well, it looks like a desert, which is not untrue, but within the deserts, there are sometimes, you know, some diamonds hidden in the ground, and the same thing occurs here. So essentially what we do is we conduct a search in PubMed, and we look for a particular condition, galactosialidosis, for example, right here. And then you can filter them by case reports, for example, and then you see that, for example, with this query here, you would identify 66 publications, and you have to go through them. Okay, now, I could talk about this for hours, but have to be a bit short. So going straight into things, how long does it take to make the diagnosis? And this is an example from the paper, from Konstantin Mechler's paper in Genetics and Medicine in 2015. Konstantin is a pediatric psychiatrist whom I worked together with on this project. And this shows you here on this graph on the right, the age of onset that we extracted from the publications, and then the age of diagnosis, which is shown here, and this slope represents the diagnostic delay in patients. And you can see that in this condition, it's a bit variable, but still substantial. Yeah. So then one particular interesting questions, I think, in a natural history program, whether you want to use it for patient counseling or to support drug development, you know, to add some additional information to the picture, you can do a survival analysis. This is pretty much Kaplan-Meier statistics with censored information indicated by the vertical lines up here, and then you can also, I mean, you define, you calculate, you model the survival based on the information that you have from the paper. And then you could, for example, divide this up by clinical subtype, for example, in galactosialidosis, storage disorders, where you have different subtypes, early infantile, late infantile, and juvenile adult subtypes, and the survival in each subtype is different, as you can appreciate from this curve here. And I think this is pretty valuable information that tells you something about the condition. And if you wanted to collect this prospectively, for example, this covers an age span of 55 years, that would take you a while, I think. So the other question, and that is, that's an interesting example also. This is one of Konstantin papers, and another paper from Konstantin and Katrin Hinterhofer, a geneticist here at the University of Heidelberg. What you can see here on this graph is the overall survival of individuals with molybdenum cofactor deficiency. You know, patients start dying after onset of the disease to a variable degree. Severe patients probably earlier, less severe patients later. And when we saw this pattern, we were wondering whether we can, identify a factor that distinguishes this, why there are these differences. And if you separate the genotypes, the published genotypes up right here, and you look at the individuals with mild mutations or sequence variants, as you say today, mild, then you can see that these patients have a pretty good survival. On the contrast, those with severe sequence variants, they tend to die earlier. So the stratification of the genotype in the survival analysis appears to have some meaningful impact. And then, of course, if you look at biomarkers, and I think the challenge with biomarkers is always, if you want a good biomarker, surrogate marker, you want to capture the overall morbidity. And then if you use it, of course, in clinical trials, you want to know whether the changes that you achieve with the treatment captures really the net effect of the treatment on the clinical outcome. And this is an example. It's also lysosomal storage disorders, a Krabbe disease. There you have elevated cerebral spinal fluid protein, CFS protein, which is probably an indication of the disruption of the blood-brain barrier. And then you can see here on these different graphs that a higher CSF protein was associated with earlier onset of disease and shorter survival. So, for example, you can see this here. These are the individuals that have higher CSF and they die earlier. And these were the individuals who had lower CSF, who had a longer survival. And you can also distinguish a difference in survival in the phenotypes. And then here you see the subtypes: early infantile, late infantile, juvenile, adult phenotype, less severe, more severe. And these subpopulation here has higher CSF protein compared to the other ones. This was published by Shoko Kamatsuzaki and Matthias Silonka from our group here in Genetics and Medicine 2019. And you can see similar patterns in other diseases, in sialic acid storage disease, for example, which you can appreciate here. This is the overall survival, and patients with higher storage excretion in the urine die earlier versus those who have lower storage excretion storing material is bad for the body. They have a better survival. And you can also look at the storage material in fibroblasts that were published in these case reports. This is all data from published case reports, and the literature shows you the same picture: high storage, lower survival, low storage, higher survival. And you can calculate these thresholds by modeling. It's another paper from Matthias Zilonka, also published in Genetics in Medicine. So then another interesting question that you could examine with this method is if you have, let's say, a neurogenetic disorder with a variable spectrum of signs and symptoms. Usually what you want to know is, are there distinct disease subtypes with a circumscribed feature of signs or symptoms, or is there a continuous spectrum of phenotype? And you can do this analysis with a technique which is demonstrated here, clustering patients with a so-called dendrogram that looks how signs and symptoms are interrelated. And then, for example, for Farber disease, another lysosomal storage disease, you could see that there was no clear-cut exclusive delineation of phenotypical features across the spectrum of disease. So it's rather a continuous spectrum of phenotype. And I think this is also important to know if you do drug development, because the heterogeneity of the disease is something you have to consider when you choose your particular clinical endpoint. Yeah, I talked about study recruitment and geographical mapping. This is an example from Thomas Slammer's paper here from our group that he published in 2019 on galactosialidosis. And this shows you countries where patients were described. And if you want to run a clinical trial or you want to do a natural history study, then you would essentially know where you want to go to. It doesn't necessarily mean that, for example, in these areas here, there are no patients, but it's not that they were not published. So maybe, you know, there's some barrier to diagnostic suspicion or access of diagnostic technologies. So talking about the limitations and opportunities, of course, you have to know that the survival data may be in part historic, and that may change over time. Supportive treatment is a very important factor that is sometimes difficult to extract from the way that papers are written and the information is available. But essentially, the survival data over time that can go into both directions. So let's say you have improved supportive care that would prolong life, but if people over time decide they choose a more palliative, medically less aggressive approach centered on quality of life, that may decrease survival time, the signal that you detect in your papers. This method, this is described in the paper. I didn't show this here, but it can detect the changing publication pattern over time if people focus on, let's say, milder or atypical cases. The epidemiological data on geographic distribution of patients, that can be, of course, of help in identifying study centers for future clinical research and drug development. Then the classification of disease subtypes, that is sometimes an issue in the published literature that may vary across case reports because the classification is not uniform in the same disease. And then you need to transparently and carefully reclassify patients published so you can match them and analyze them. And there you have, of course, you have to be transparent. The main issue is that the case reports often focus on a specific aspect of the particular disease, depending a bit which journal they are written for or what the interest of the researcher was. And therefore, standardized quantitative description of softer variables, quality of life, development of motor skills, speech development, cognitive function, seizures, joint mobility, skin nodules, or whatever, is these are, of course, subject to ascertainment bias and missing data in your analysis. Published laboratory data are often not analyzed in a central laboratory, so you somehow have to pool them in order to allow some comparative analyses. this, of course, introduces some statistical noise. This is an example about different ways you could do. It's almost like a consumer report. What your pros and cons of different ways of doing natural history studies are. So the method that I had presented was quantitative retrospective natural history modeling. Then you could also do a prospective natural history study, which means you write up a protocol, you submit it to your IRB, and then you enroll patients prospectively. Or something within between a little bit is retrospective natural history studies, for example, on chart reviews, which means you go to the centers where patients are treated and then you collect the data that are there. I think the advantage of the method that I had presented is that the time to availability of hard endpoints, in particular survival, is pretty fast in quantitative retrospective natural history modeling, because it just captures the information that is published and available, and you do this prospectively, and you have a disease where, let's say, patients die in their 20s, 30s, 40s, or 50s. Fabry disease is an example. It takes you very long to get these data. On the other hand, defining softer clinical endpoints, as I said, are difficult to capture in quantitative retrospective natural history modeling because they are sometimes they're just not there, not published. That is easy in a prospective natural history study if they are part of your protocol and you're particularly looking at it. And in chart review, it's probably somehow within between. Defining biomarkers is difficult from the literature data because they may not be there. Easy in prospective natural history studies because you particularly look at them, you make them part of the protocol, and of course sometimes you have a central laboratory which gives you some more cleaner laboratory assessments. The logistic burden, though, in quantitative retrospective natural history modeling is relatively low. I mean, you need an internet connection. As I showed you, mine for the seminar didn't work in the beginning. I'm sorry, but usually it works. And you need access to the medical literature, to PubMed, and get the papers. And then, of course, you need some dedicated people, people who are interested in the disease, who, you need a good biostatistician who understands clinical questions and combines that with analyzing skills. It's always a pleasure if you work with statisticians who understand clinical diseases. And I was very lucky in my career that I've worked with these people. Sven Gabade, for example, he's one of those. Right, the logistic burden, of course, in a prospective natural history study is huge. You have to write the protocol, you have to set up the centers and do the analyses, and it's a lot of traveling, data monitoring, whatever. In retrospective natural history studies, it's, I'd say, within between the two of them. You wouldn't enroll patients because you go to see the charts, but you still have to go to the centers and collect the data. So now a practical example where this approach was supportive for drug development, I would say. There is a condition, as said in the beginning, a metabolic epileptic encephalopathy, which is called molybdenum cofactor deficiency type A. Due to a genetic mutation, patients are not able to synthesize molybdenum cofactor, which is a biochemically important factor for certain processes in the body. If you cannot make this cofactor, you get sick, and there's a metabolic block here in molybdenum cofactor deficiency type A. This gene is mutated, and you cannot make this compound, which is called CPMP. But fortunately, you can make this compound artificially and substitute it. And this is the story of molybdenum cofactor deficiency A drug development. There was a drug approved in February 26, 2021. And what was our relationship to that program? Katrin Hinterhöfer, my genetic colleague, and Konstantin Mechler, who was my MD thesis student at the time. They looked at the published cases of molybdenum cofactor and worked out the differences that you could distinguish survival by stratifying genotypes. If you go and look at patients with mild genotypes, they have a pretty good survival. With severe genotypes, they die earlier. So as we wrote, the severity of genotype assessed by in silico prediction and further classification explained the survival in molybdenum cofactor deficiency. And we propose that they may therefore be considered as a confounder for the outcome of therapeutic clinical trials requiring adjustment in the clinical trial design or analysis. There were patients treated with CPMP. And this is a chart, this let's say an image that is taken from the FDA, the review of this drug development program. And these are treated patients here, and their clinical course, their outcome, the survival is compared to untreated natural history controls that were collected separately. And then you can see a difference in survival between the treated group and the genotype matched natural history set. So these are matched for genotype in the analysis. But this image, I like this image because it shows you a lot of interesting aspects. So first of all, it shows you that drug development is somehow a global endeavor. People have to work together, exchange knowledge, collaborate, and it all started here in the lab of Dr. Schwartz in Cologne, who was working on CPMP substitution in MoCo type A mice. So he was working, if you want to say, so in a preclinical model and published that. And then it happened, this was in 2004. And the approval was in 2021. I don't know who works in the same company for such a long time, but these are the numbers. So in 2010, Alex Feldman, who is a pediatric cardiologist from Germany, at the time he was working at Monash University in Australia, and they diagnosed a baby with MoCo deficiency type A and somehow established the connection to Günther Schwarz's lab, who sent them CPMP. They treated the patient and saw a beneficial effect. That was the first in human treatment on an emergency protocol, because you've seen the survival curves that I showed in the beginning. The outcome is fatal. And they were pretty much encouraged by that. And then what happened later is that people started treating patients. It's a rare disease, so patients are occasionally being diagnosed. And there was a publication by Bernd Schwan in 2015 from the UK, where he published, or the group published, data on an uncontrolled multi-center open label study. So this is not a randomized controlled trial, because the condition is rare. Patients are living in all regions of the world, but he just published the experience with the treatment and documented some effects. In 2017, we published our natural history study and had proposed the genotype stratification as a sort of clinical regulatory strategy for drug approval. The FDA approved the compound in 2021, and the natural history study data set that supported approval, that was published by Dr. Spiegel in 2021 in the Journal of Inherited Metabolic Disease. So it takes time, and it's a global effort, and you need some dedication. But it's a nice story. So let me conclude that quantitative retrospective natural history modeling can provide fast information on hard clinical endpoints, survival, diagnostic delay, with a relatively low logistic burden. Now, the choice of method to quantitate the natural history of a disease, for example, in a particular drug development program, that may be driven by the research question overall and the understanding of the disease in the particular condition. So are you looking at organ manifestation, disease progression, progression, I'm sorry, the nature of endpoints and the feasibility of assessments. And it overall may encompass a combinatory approach, which includes quantitative natural history modeling based on published cases as supportive data. Perspectives, we are expanding this method now into epilepsy genetics. This is Steffen Service team, a very esteemed colleague of mine in pediatric epilepsy here at the University of Heidelberg. These are two papers we published, ongoing projects. And this is an overview of the literature, and you find some of the examples that I showed on the slides in these papers. And this concludes my presentation. Right. And I should be back on screen, I hope.
You are. Thank you very much, Dr. Ries, for the excellent presentation on this unique natural history approach. I see a couple of excellent questions in the chat, but we are a little bit behind due to the technical difficulties. I believe the speakers do have the ability to go back and actually answer the questions within the chat. So maybe I can ask that if any of the speakers are able to do that, and maybe can be able to address some of the questions that we've not been able to get to during the presentations. But at this time, I'd like to move into our panel discussion. It's supposed to end at noon, but due to the technical difficulties, I'm told that we can take until 12:15 to make sure that we can have an adequate discussion on this topic. So at this point, I'd like to invite Dr. Damian, who is the Senior Director of Systems Modeling and Translational Biology, Computational Sciences, Medicine Design, and R&D at GlaxoSmithKline, who will be moderating the session, as well as all of our panel discussion participants to turn their cameras on. Thank you.
Hi, everyone, and thank you very much for the invitation to moderate this fantastic session. Hope you can see the slide, my screen. Can I get a confirmation?
Yes, we can.
Brilliant, thank you.
There's a bar down below, but otherwise we could see it.
Okay. So I wanted to take this opportunity first to acknowledge our panelists. So aside from our speakers so far, Dr. Zineh, Dr. Lee, and Dr. Ries, we are also joined here by Stephen Chang, who is the President and CEO at Immunetrics, and by Vernon, Associate Professor of Genetic Medicine, Department of Genetic Medicine at Johns Hopkins University School of Medicine. And we also have the session chair, Bob Schuck, present in the panel. So we have quite a number of questions at this point, and I think it's best to just get started with them. We have prepared a few questions for the panelists, and we're going to be interlacing them with questions from the audience. So let me start with the first question, and this is for all the panelists. It has been already established that in rare diseases, the variability is and the lack of patients would mean that the trial size required to demonstrate the treatment effect, particularly if that treatment effect is not huge, is very challenging and very challenging to recruit. So the question is, how can QSP help? And is it what do you see the role in accelerating the rare diseases cure through QSP? What are the rare disease drug development needs you would like to see the QSP field focus on. And perhaps stemming from the talk by Dr. Zineh earlier, if we're looking at the biggest challenges that we have in rare diseases, is there a need actually for a step change? We see that the challenges are out there. They are enhanced by the application in rare diseases. Do we need to do something different to accelerate its use in rare diseases? So I don't know if perhaps Dr. Zineh, you may want to start the discussion.
I'll say a couple of things just to lead off. So I think the biggest, one of the biggest potentials for a QSP is in making sure that the studies are as least noisy as possible, right? And so when we think about being able to tease out a signal, being able to determine that a treatment effect actually exists, if it does exist, right? There are three maybe sources of noise that I could think of. One is this variability of variability in disease progression, which we've talked about. And so thinking about how to enrich a clinical study, a clinical trial, so that you're minimizing that noise around disease heterogeneity would be one strategy. The other thing we haven't really talked about is a kind of predictive enrichment. How can we, these are very targeted therapies, but is there a way to understand sort of baseline covariates and their relationship with response? And oftentimes that can't be done by clinical data, because as we talked about, these are generally unprecedented targets, but QSP may be able to help at least inform that question of predictive enrichment. And then not all of these rare diseases are done as single-arm studies. They can be done in with placebo comparators. And so, another I guess source of noise, if you will, is the placebo response. So is there a way to get a good understanding of what the factors that are contributing to placebo response might be? So those might be areas in which, at least when we're designing these small trials, those heterogeneities could be considered and minimized in order to really amplify the signal around the treatment effect if it exists. The other thing is around, I think I mentioned with the survey data and kind of where I see the biggest opportunity, biomarkers. Oftentimes we cannot access the target compartment of the disease, and so is there work that could be done to help identify some soluble biomarkers that might increase our confidence that if you get some traction and movement on that biomarker as a result of a therapeutic intervention, that there's going to be a relationship with clinical outcomes that we may never see, because it may take a very long time for a disease, the end sequelae to manifest. So those may be the two kind of big opportunities that I see.
Thank you. Any other panelists that would like to? Yeah, Steve and then Hillary.
Hi, Steve Chang. Yeah, I think that sort of the big challenge is really understanding the implications, even from a regulatory perspective, of the fact that obviously in these illnesses, we're trying to settle on smaller ends. Statistics starts to play a more limited role because you can't really look so much for statistical significance. That then yields compromises. Those compromises are also inherent in QSP, because you can't build a QSP model that's devoid of data, right? Well, you can, but you probably don't want to trust that very much. And so the challenge is, where is that new bar? Because the bar for traditional trials and QSP is set where they don't have these constraints. How do we operate in a domain where there almost always will not be enough data? What is the expectation and what's sufficient? So it's almost a question back to regulatory. How do you address that issue? They're trying to address it with the actual trials themselves. Now, how would you look at that in the face of QSP?
Dr. Lee wants to mention something here, especially around sort of the, being proactive around
Yeah,
confirmatory evidence.
That's exactly right. Thank you, Issam. This is kind of where I was going to hop in. I think the challenge in rare disease drug development is you can answer these questions during the trials as much as you can in more common disease development, because you don't have the multiple phases and the luxury of going back and refining after you've interpreted the data over and over and over. So I think where, with QSP being informed essentially by its priors, right, the strength of your prior evidence, you really have to do due diligence in gathering as much and as sort of as much data as possible about the natural history and as much data as possible about translational science that informs both whether it's your selection of a surrogate endpoint or confirmatory evidence. Because as I said in when I spoke first, that the majority of rare disease drug development trials that are approved follow establishing substantial evidence of effectiveness through one adequate and well-controlled trial plus confirmatory evidence. And that confirmatory evidence is very grounded in the strength of the translational science that informs that. So I think it's just, you have to do as much as possible to strengthen your priors to inform your QSP when you're going there. Those are my thoughts.
Thank you very much. I saw Dr. Vernon, you had your hand up.
Yeah, so one thing that I wanted to chip in was the importance of thoughtfulness about prospective natural history collection and trying to tie in some of the data that's collected, endpoints and questionnaires, to things that would be included in an eventual clinical trial. And I think part of that really is being able to not only generate a really nice natural history comparison group, but being able to use patients as their own controls over a longitudinal time. And the other thing I think I wanted to chip in, which would be really helpful, which would be to be able to use natural history comparators, not just to support clinical trials and patient selection, but actually as a confirmatory study to gain approval, or at least prospective approval for drugs in which we really can't repeat clinical natural history clinical trials in an ultra rare disease. That's something I haven't quite seen yet, but I think that would be incredibly helpful moving forward.
Thank you very much.
If I can maybe jump in quickly, just to piggyback on maybe the last couple of comments, being deliberate about natural history studies, completely agree with that. And Dr. Lee mentioned, the translational science and really understanding that. I mean, one thing that we've been trying to do within our group is really encourage companies to be deliberate in thinking about their biomarker strategy early in drug development. So because a lot of these programs end up relying on that confirmatory evidence paradigm, really thinking through what biomarkers need to be collected, when they need to be collected, what media they need to be collected in, is it blood, urine, CSF, thinking about all those things early in your phases of drug development so that you have that in as many patients as possible and can have that translational science component be as informative as possible on the back end, because it makes it much more difficult when you're trying to, figure out what the confirmatory evidence is late in drug development, as opposed to when you've collected the data that can help serve as that body of confirmatory evidence. I think that's one thing companies can do, and modeling can play a big part in that as well. And again, as others have pointed out, you need the data to be able to model. So being deliberate about your biomarker strategy early, I think, can be very important.
So if I may, I'll switch the discussion to the second question there. It's been identified through a couple of surveys that building trust is one of the key challenges in enabling QSP to reach its potential. But this challenge is actually quite magnified in rare diseases, due both because of the number of diseases, sparsity of data. And the question is, can you comment on how to get trust and buy-in from QSP modelers, from biologists, from clinicians, from regulators, from other non-modeler stakeholders?
I can just lead off very quickly. I mean, I think this really underscores the importance of working together in the pre-competitive space. There's a number of consortia or pre-consortia that are out there. It sort of speaks to the efforts to try and gather data collectively, efforts such as the RDCA DAP, where there's a number of other efforts that are out there to do that. I think that that trust is is really something that you gain working in the pre-competitive space in order to sort of really gather as much data as possible to inform drug development.
Thank you. Any other thoughts around these questions, Dr. Zineh?
I mean, I think it depends on what we mean by trust. You know, I think this sort of QSP and mechanistic approaches are a lot more conceptually appealing and intuitive to non-modelers. I mean, they get a pretty good grasp of the biology, and sort of, you put a cartoon in front of a, you know, clinician describing the sort of in front of an immunologist describing the immune system, they get it. So they trust, I think trust in terms of conceptual willingness to engage on the issue is there, and maybe even easier than in some of the more empirical strategies that have been around for much longer. I think if we're asking people to make decisions, you know, solely based on model output, that's a tall order, you know, and so this goes back to, we calibrate our trust in the model based on what's the intention for the model, how does it fit into the overall sort of picture, and what is the consequence of being wrong in that trust. You know, if we're talking about clinical trial planning, the consequence of being wrong is that ultimately we'll have the clinical trial that gives us the readout that we can make our decision on. If we're talking about completely, you know, replacing a clinical trial, that's a higher bar. And so I think when we talk about trust, we really need to put this in a more nuanced discussion around what is the utility of the model output, and how does it fit into a totality of evidence consideration.
So let me just say that when I thought about this particular question, your talk actually, Dr. Zineh, sort of illustrated this as well. There is a question of trust with regulatories and biologists and clinicians and so forth, but there is also the trust among modelers, wherever they are. And if I take there were surveys out there that were showing that even if I take a model from a colleague or from a CRO, there is a long period of time that it takes for, you know, myself as a modeler to trust that. And I wanted to flip that question in terms of the regulatory process, because what we see on the industry trying to trust something that's out there published or from a CRO before we can leverage for our decisions, from a regulatory perspective, you need to do the same with models that are provided to you by different sponsor companies. And in the context of rare diseases, this becomes much more challenging because of the throughput of the number of such models that need to be addressed. Can you perhaps give a perspective on this?
Okay, thank you. That clarifies a bit. I think there are a couple of from the regulatory side, you know, this idea. I'm going to say openness to, or willingness to consider, you know, MIDD strategies. One of the things that has been very helpful as an outcome of the MIDD program is that we have a sort of centralized, coordinated effort that allows decision makers across different therapeutic areas to understand precedent. So one of the major drivers of reluctance is, well, maybe I've never seen a QSP model used for this purpose in my therapeutic area, but maybe we have 10 other examples where that's been the case in other therapeutic areas. So I think sharing the experience across different use contexts is very important. That's sort of one issue. The second kind of dimension, not to belabor the point, is sometimes we will have proposals to use QSP or other models, and it's actually not clear at all what the purpose is. So it's sort of, you know, let's put this into the regulatory submission, let's test the water out and see what the reaction is going to be. But there's not actually a clear context of use. There's not a regulatory decision per se that we're trying to influence. And so it's not so much an issue of trust, if you will. It's sort of like, well, what are you asking me to react to when the actual application of this, you know, model has not been clearly articulated? So I would say those two those two aspects of just getting oriented to what is being proposed, I would say probably accounts for the majority of the reluctance, if you want to call it that.
Thank you very much. So I wanted to switch to one of the questions from the audience from the chat. How do you assess the use of real-world data in ultra-rare diseases, especially in the context of single-arm trials? I don't know, perhaps.
I can take a stab,
Sure.
just because I don't think, and I'm actually going to try and find it and put it in the chat. One of the biggest things we've learned through Leader 3D, we actually have a tremendously robust real-world data program at the FDA that puts out an enormous amount of information on how to utilize real-world data, how to collect good real-world data, how to then potentially build the case of utilizing real-world data in the context of real-world evidence, plus the recently released external controls guidance that they just did. And when I pause, I'm going to find the link and put it in there for everyone. But I think there's a number of considerations when it comes to single-arm trials, and the key context, it goes back to, you really have to have a well-characterized natural history for a disease in order to be able to potentially consider single-arm trials. There's also the context of, it is most helpful when the endpoint is an objective one that is clear to measure. It is also very helpful when the treatment effect is not modest, when it's something that the treatment effect is large. There's a lot of just sort of basic considerations that go into whether or not it's even possible to consider a single-arm trial in the context of a rare disease. But I'll put the link for that, as well as the external controls guidance, in the chat.
Thank you. So I'm wanting to move to the question number 3 there on the screen, and it sort of refers back to your talk, Dr. Lee. One of the conclusions is rare diseases are challenging engage with the FDA early and often. So basically the question is, what are mechanisms to engage with the FDA on QSP beyond the MIDD program or individual sponsor meeting?
Yeah, this is an oft asked question. I mean, we try and do a lot through Cedar Arc this year in terms of engagement, just more broadly, and sharing learnings, particularly about, new methodologies and innovative methodologies and how the FDA is thinking about those and using those. But other mechanisms for more individual engagement, if it's regarding a drug product, we do have pre-IND meetings that occur prior to your IND that you're developing, and those are helpful. We also have a guidance on how to engage with the FDA around pre-IND meetings out there. We additionally have the interact meetings, which are kind of a pre-IND. Again, this is when you have a drug product, but you're really in the early stages and you want to talk about something perhaps particularly innovative to incorporate in your IND in your drug development program. So those are two very good ways to engage regarding a product. And then Issam, I don't know if you wanted to add a little bit more about the role of like a CPEM or those types of
pathways just to consider. One is we do have critical path innovation meetings. These are non-regulatory kind of pens down meetings where they're essentially exchanges to understand what the issues are, what the sort of science is telling us, or what we think the science is telling us. And it's a nice also triaging point if there's need for further kind of binding regulatory discussion. We can sort of direct people that way. The other program is the Fit for Purpose initiative, which is, we realized think of it as almost a biomarker qualification program, but for models. We haven't had QSP models come through there, but we have had disease progression models, placebo models, statistical models come through there. And essentially it's a way to have an interdisciplinary team, including clinicians, statisticians, pharmacometricians, clinical pharmacologists, et cetera, evaluate sort of the claims that are being made by the model and how it might be by the submitter about how the model might be useful in drug development. And then those are made public. So that's another mechanism. And then, you know, at least in our office, we're very interested in learning. Dr. Bai, Dr. Schuck, we have a visiting sort of lecturer series. And so if it's a sort of an interest in education, methodology, that sort of thing, you know, they would be the points of contact to engage a bit further on that from an educational standpoint. And that's broadly, that's open to individuals beyond just our office as well. So those are just three that come to mind. Thank you very much.
I wanted to go back to some of the questions from the audience. So this is for Markus Ries talk. So in your experience, how much does the patient environment play a role in the rate of disease progression relative to the underlying patient genotype or mutation?
Yeah, this is an excellent question because for a disease phenotype, of course, the genotype is decisive, and then the environmental factors, concomitant treatment, and there are all kinds of confounding factors that you have to consider. We per se, I think, cannot quantify the effect of the environment because it's not part of the publication. But I think if you have a disease where the genetic impact is so huge, it probably doesn't have such a strong effect. What you could do in your model to increase confidence, you could do some sensitivity analysis. Let's say if you have a natural history program that is based on quantitative retrospective natural history modeling, and the collection of prospective data and the signals that you see are consistent, that gives you more confidence, I think.
Thank you very much. Dr. Vernon, do you wanted to add anything to this question?
No, the only thing I might want to add is, particularly with a lot of the diseases that I work with, intercurrent illness causes a vast acceleration of the progression of disease. So in that sense, those kinds of environmental influences really change how we track events, actually. And so this is sort of a different way of looking at environmental exposures, but certainly intercurrent illness really affects a lot of rare and ultra rare conditions, metabolic diseases.
Thank you very much. So I'm going to try to move to the fourth question here. So can you comment on any parallels between the application of QSP and PBPK in rare disease drug development and compare that with the pediatric drug development? Are there any parallel, are there any learnings from one to the other? Dr. Schuck, maybe you can start, or I don't know who would like to provide a view on this.
As a non-model, I don't think I have a ton to add, and maybe direct the question toward Dr. Vernon, Dr. Chang, but I mean, certainly a lot of, or probably the majority of rare diseases are actually pediatric diseases. So there's certainly a parallel in that light, in that one of the challenges that we're often trying to accomplish is extrapolating dose, extrapolating efficacy findings, extrapolating safety populations from the ages that were enrolled in clinical trials down to younger ages that may not have been represented or weren't represented very well. So I can see a parallel from that perspective, but would defer to some of the other folks that have more experience with the modeling components to answer.
Any other thoughts, comments?
Just, I mean, I think the thought exercise is the same. What do we know about, oftentimes, as Bob says, when we're doing in the pediatric space, it's largely about extrapolation. And so what do we know? And that comes down to what do we know about the disease? What do we know about the disease, and benchmarking it against what do we know about the disease in adults, whether that's the pathophysiology of the disease, the natural history of the disease, also the determinants of drug disposition, those might be different when we're looking at kids versus adults, but the thought exercise is still the same, kind of a mental map of our understanding of the biology and relevant processes, and then contextualizing it to the question of what are we extrapolating? Is this an efficacy question? Is it a differential safety question, which is often the case between adults and kids? Or is it, is it one of dose optimization? So I would say very, very analogous scenarios.
So I wanted to bring the last question there, and it's very much related to a question actually from Dr. Chang. The question on the list here on the screen: if we extrapolate the definition of a rare disease to a single individual, can we consider the application of QSP to rare diseases as a step towards its application to personalized medicine? And the connection to that also is with the question from in the chat from Dr. Chang, with the concept of N equal 1. What are the thoughts about how this might influence or apply to QSP and modeling? Maybe Dr. Chang, I'll give you the chance to answer the question that it's posed here on the screen, and perhaps somebody else could address the question you posed.
Yeah, I think that the use of natural history is very interesting. So in rare disease, getting models to maybe QSP models to recreate the natural history in a literal sense is certainly a form of calibration that can help reduce some of the variability and inform the model in manners that would allow it to compensate for the fact that there's less sample size data, but there's more longitudinal history data. So I think that that's certainly a different way of thinking about the problem with respect to how QSP traditionally is built in other illnesses. And I think that that is something that, if it's made more available, because a lot of these, as was mentioned in the pre-competitive space, the collection of that data, et cetera, how available is that to industry as opposed to academics for research? That's something I don't know about, but that's a follow-on question from me.
So anybody wanted to comment on the N equal 1 question that Dr. Chang has posed?
The only thing I might want to add is when you're dealing with ultra rare conditions, sort of on a day-to-day basis in a clinical setting, almost everything you do is an N of 1 test. And so I think from that perspective, it's something that actually feels rather comfortable and tenable, understanding a disease quite well, intervening with a patient and seeing, does this contradict what we would see in the natural history. But I think from a quantitative perspective, obviously we need a lot more data than just our day-to-day experiences. But again, I think it's something that a lot of people dealing on a day-to-day basis with ultra rare diseases would be quite comfortable with managing.
Thank you very much. I wanted to end with one last question from the audience. So does the FDA sponsor for a natural history study or a QSP model for a specific rare disease?
So there are natural history grants available through the Orphan Products Group, and there have been for several years.
Thank you. So I wanted to thank very much all the panelists. We are a little bit over time, but I think that's kind of the time we wanted to finish the panel. I wanted to thank all the panelists for their thoughtful answers to the questions raised here. And I think we have a break for the next maybe 45 minutes or so.
Yes, thanks a lot, everyone. We'll be back at 1 to start session 2.